Hello everyone. So I'm Antoine Felden. I'm going to talk about the research we do on bee pathogens at Victoria Uni in Wellington, and especially about virus-virus interactions in the honeybee parasite varroa-destructor. Oops. So this bee that you can see here on the screen is not doing very well. You can see three varroa mites on its back. Varroa-destructor is a globally invasive species that pretty much affects all the honeybee populations around the world. Until earlier this year, we could say that Australia was the last significant landmass to be devoid of varroa, but it's not true anymore. Fingers crossed they can eradicate it. They're still trying to do that. And it's a parasite, ectoparasite, that is associated with hive mortality around the world. Represent 5% of all New Zealand hives lost to varroa in last year, which is a staggering 110,000 hives. And that number is still increasing over the years, so it's becoming more and more of a problem. That represents a huge cost for beekeepers, both in terms of direct losses, but also in terms of expensive treatments against the mite. Another big threat for honeybee, and you can't really see it on that picture, but you can see the symptoms, is the deformed wing virus that causes, as the name suggests, deformed wings, and also all sorts of cognitive problems and overall lower survival of honeybees. These two together are the main cause of hive losses around the world. And they work in a kind of a synergetic way. So varroa-destructor lodges itself generally on the side of the bee. You can see it. Oh, yeah, there's an arrow here, you can see it. And attach itself on the side of the bee and repeatedly feeds through these feeding holes here at the bottom. By doing that, it vectors deforming virus to the bees, and also by feeding on the fat bodies that function as that have an immune function in insects, lower the immune function, and potentially increase the infection by deforming virus. So it's kind of a recipe for disaster for the bees, having these two threats working together. So this project started by doing quite a simple survey using RNA-sequencing of honeybees and varroa-destructor around New Zealand. And you can see we have a bunch of samples there, and viral loads measured as a number of reads mapping to deformed wing virus or other virus. The main virus that we see in bees on the top plot in yellow is deformed wing virus. There's a few other different viruses, but in terms of significant loads, it really is deformed wing virus that is mainly affecting the bees. On the bottom plot, which is the viral loads in varroa, we can see also significant loads of deformed wing virus, and also consistently this other virus shown in purple called varroa-destructor virus 2, VDV2. Get used to the acronyms, deformed wing virus and VDV2. They'll appear on the plots quite a bit. So yeah, that was just a little survey, but that started quite interesting prospect. So first, the relationship between deformed wing virus in varroa and in bees. That's quite a known relationship. The more deformed wing virus you have in varroa, the more deformed wing virus you have in bees. So that just highlights this interaction between varroa and deformed wing virus that affects the bees negatively in terms of deformed wing virus loads. That was quite a known relationship, obviously. What was more interesting is that, so if you look at just the first plot there, that it shows the VDV2, the varroa virus loads in the x-axis and the deformed wing virus loads in the y-axis. And you can see this very strong negative association between the two, which suggests that these two viruses might be interacting in some kind of way and negatively affect each other. We don't know in which way, if it's one inhibiting the other or just a competition mechanism, but there's definitely something happening there. And most interestingly for honeybees, whenever you had high VDV2 loads in varroa, you never found high deformed wing virus loads in bees. So that reflects the relationship within varroa with flow-on effects on honeybees. So that's a project that just that just received some funding from Marsden, which I'm really happy about, studying the viral interactions within varroa, especially in this case between VDV2 and deforming virus, and how that affects viral loads, deforming virus loads in bees, and ultimately colony fitness. So there's not much genomics in there, but I'm going to get to that because that's part of the project. I'm going to expand on that. So I'll be starting this project in March next year. So deforming virus is a single-stranded positive RNA, positive sense RNA virus, and these kind of viruses are characterized by really high mutation rates by design. And that has a consequence of they typically exist as quasi-species characterized by mutant clouds. So a bunch of different variants moving together through hosts with tiny or larger variations. In the case of deforming virus, in the sequence, in the case of deforming virus, we have two master variants, deforming virus A, that is present in New Zealand, and deforming virus B, that is not present in New Zealand. But that is slowly overtaking, or at least increasing in terms of prevalence around the world. Deforming virus B is slowly increasing in terms of prevalence. And that is thought to be due to this symbiosis with varroa. So in this fish analysis down there, that's a recent paper, that showed that, so that's a cross-section of a varroa gut, and using probes specific to either deforming virus A or B, they showed that although deforming virus A was detected using qPCR, so it was present in some extent, it was not able to infect the cells of varroa. And on the other hand, deforming virus B was able to actually be found within the cells, which suggests that deforming virus, this is quite controversial at this stage, suggests that deforming virus A is not able to infect varroa, but deforming virus B is. Another line of evidence for that is that they sampled viruses in varroa-parasitized bees, and they did find a lot of deforming virus B, but when they isolated these viruses and passaged it to virus-free bees, then there was somehow a sequence shift, and they wouldn't detect any more deforming virus B, but they would detect deforming virus A. Still very controversial. I'm still not sure I believe that story, but they explained that by the fact that distinct deforming virus variants may be specialized to infect distinct hosts, and that host switching may act as a short-term evolutionary bottleneck and be the cause of that switch between the two master variants. On top of that, we did a little study, another survey in 2020, looking at deforming virus, this time not looking at different variants, but looking at deforming virus presence in a bunch of insects that were found around apiaries in New Zealand. And you can see that deforming virus was detected in a bunch of organisms that were not bees, so from spiders to cockroaches to flies and other hymenopterans and orthopterans. So deforming virus has a really wide host range, which has two implications. First, there's many chances of it being introduced from other sources than honeybees. So honeybee exports stopped in New Zealand. I wasn't able to really find the exact date, but a while ago, at least a few decades ago. But deforming virus and other variants of deforming virus could be introduced to New Zealand through other hosts, for instance. And another consequence of this large host reservoir is that it can promote mutations, which could lead to accelerated evolution. So the thing is, so we don't have deforming virus B, the type that is supposedly only infecting varroa in New Zealand. We only ever detected deforming virus A, but yet there's still a strong association in terms of mortality in bees between varroa and deforming virus in New Zealand. So our working hypothesis for this project is that high mutation rates in deforming virus as an RNA virus led to the emergence of distinct variants in New Zealand, and with the variants in New Zealand exhibiting deforming virus bee-like properties, as in being able to infect varroa, where this didn't happen and maybe took another evolutionary trajectory in terms of genome evolution. So to explore at least these questions, I'm really excited to use reverse genetics methods to resolve pathogen evolution and evolution of virulence. So the idea of reverse genetics is to first get an idea of the genetic diversity of deforming virus in New Zealand, and we'll be using nanopore sequencing for that with our collaborator in Australia. And then to insert the full sequence of the viruses that we found in New Zealand into a plasmid to be able to experimentally inject this plasmid into the host and see, well, first see if the host develops an infection depending on the variant, but also to study the core of this project to actually look at viral interactions that we're able to control and manipulate in the lab. So yeah, not many results because that hasn't started yet, but really looking forward to do it. And I'd like to thank the whole Phil Lester Lab, collaborators in Australia and New Zealand, and a special thanks to Invasive Hubs for getting me here. Thank you very much.
Thanks, Antoine. Question right here.
Hi, yeah, I'm curious, you had mentioned you found the deformed wing virus in a lot of different species, I believe. Has there been any attempt to kind of put that into a tree and see how they're related and how the A and B are related to each other in the context of that larger diversity?
No, we did that using qPCR that were not actually variant specific. So we know that it's some kind of deforming virus. We also looked at replication, and in some cases there was 2 + sign that meant it was replicating in the host. But we didn't look at variants in this case. We were not really aware at the time of this whole variant controversy. So, yeah.
Cool. It just seems like that would be really interesting.
Yeah, that
to
would
see
be
what
very
that
interesting. But
diversity
and
looks
going
like.
back to these samples would be good. But yeah.
It sounds like you have a lot to do already, though.
Yeah.
Thank you.
I'm just, oh, no.
Hi, thanks, lovely talk. I was wondering, it's sort of related to the host response, is the phenotype all the same across all those different organisms, or are you using quite a broad brush when you talk about deformed wings as a phenotypic response to that virus?
It's also a very controversial subject. People argue a lot about whether deforming virus A or B is more detrimental to bees or not.
Yeah.
There's studies that say yes, studies that say no, so I can't really answer your question in a definite way.
Any other questions? Aha. How many bees do you have to screen before you can say a virus is absent?
That's a good question. That was a big criticism on the marathon too. Well, it's not just us that have been looking for it. MPI has been doing a large-scale study across the country with thousands of samples, hundreds at least, and they didn't find it. So that's true. You know, we can never know. You know, if it's very rare. But given that it's slowly replacing the other genotype in the rest of the world, we would expect that by now it would be quite prevalent if it was here. But it's still possible that it's here and no one has found it. Yeah, I'll keep looking for it.